Good morning, everyone. Today, the students are still very less. I don't know the reason. Anyway, that I need to start our class today, October, November 16, variant 4.3. And this was, I gave you the PDF, that document yesterday. So I'll show you on the screen like yesterday. I'm going to show you this on our screen. You can see this thing through the screen. See, girls. The first page, the second page. Yes, miss. Ah, 6 participants. Okay, my students are coming. Okay, Maisha came. Okay, girls, we are going to start. Look at the first question. Yes, miss.
Yes. The variant, the last variant, we were doing February, March 17th.
I told you, let us finish the 16 next week, inshallah. I'll give you the PDF of 17. And how many, that February, March 17, how many questions we didn't do, I will do next class. Let me finish.
That's it.
That 16. And tomorrow also you have class with me. So do one thing, that because everybody is not ready with the February, March. I didn't inform them. So tomorrow let me.
It's the variant we did with the guys.
I know, but I didn't inform the girls, so they're not ready with the February, March. So I will give the PDF question today.
Has the paper.
All of you are. Do you have this paper with you right now?
Not now, miss.
Yeah, that's why I'll send you the PDF file tomorrow. So tomorrow I'll solve February, March 17, right?
Okay, miss.
Okay, so Anika, yesterday the question, Oxin question, you have that sent in my WhatsApp. I told you Oxin questions was a bit more explanation you need. You just listen to me. This answer is for Anika. Oxin made in shoot tape from tape.
I'll just take out the question.
Sure. Just look at your answer and what are the points that you have missed, just write it. Oxin made in shoot tape from the tip. Oxin diffuse and collect in the lower side of the stem. Oxin distributes unequally on the stem. Where oxin accumulates, it stimulates the cell elongation compared to the area where the stem where oxins are not or less accumulates.
Miss, what are you talking about?
Miss,
this is for Anika.
Repeat from the part, like oxin accumulates. What's the thing after accumulation? I didn't understand.
Oxin distributes unequally on the stem. Where oxin accumulates, it stimulates the cell elongation. Where oxin accumulates, it stimulates the cell elongation.
Okay, then.
Compared to the area of the stem where oxins are not or less accumulates.
Compared to the area of stem.
Of a stem where oxins are not or less accumulates. So, oxin accumulated stem part will grow faster.
So, oxin?
Accumulated part, stem part. So, oxin accumulated stem part will grow faster, which causes curvature. Okay, now everyone, girls, this was the Anika's problem that she informed me yesterday. So, let's look at question number 1. By looking this picture, you can understand an in vitro fertilization or IVF procedures are going on. So this they have outlined this figure. You can understand the cervix, the uterus. These all are clear here, right? So here, look at the ovary. where the egg cells are producing, right? So through the injection, they have taken the egg cell, they have kept it in the petri dish where the sperm cells are there. And the zygote forms, the embryo start forming outside. So they are taking through the injection, this one of the embryo, and embedded in the wall of the uterus. So could you please, you know the name of A, right?
Cervix.
This is vagina. The B is oviduct or fallopian tube. Look at the B. You can write oviduct or fallopian tube. And the D is sperm, right?
Petri dish. Sperm. Okay.
Or you can write the male gametes also. And now if I'm going a bit down to our next question, see, name the hormone that state the purpose of the syringe. The syringe they have used here to remove the egg cell from the ovary, right? You can see that syringe C, they're asking, not syringe E. Syringe C, the purpose to remove the egg cell from the ovary. Look at the diagram. The diagram, it is telling you this structure is working like that. So syringe C is using to remove the egg cell from the C. But if they're asking syringe E to take the embryo from the petri dish and to insert it in the uterus, you could write like that. Whatever you are saying in the diagram, you will explain exactly same way. In question B1, name a hormone that would be injected to stimulate egg cell development.
Progesterone.
FSH.
Follicle stimulating hormone, not progesterone.
Hormone.
Follicle stimulating hormone, because it's egg cell development. It is progesterone will be in the lining of the uterus, or luteinizing hormone also you can say. And next one, state when during the menstrual cycle the hormone should be injected. This follicle stimulating hormone.
Ovulation.
Before the ovulation, within 1 to 10 days, right?
Okay. Yeah. Before the ovulation.
Now draw an X at the position where embryo should be placed. Any place. Look at the arrow, girls. Here, this is the endometrium wall. This is the endometrium wall. In these two places, any place you can draw a X to show the place where the embryo can embed it. Next one.
The Xmas?
Sorry?
Where should be the Xmas?
Endometrium wall.
Oh, okay.
Endometrium wall, and their diagram is clear. They showed in different shade this endometrium wall. Outside, I told you the outer layer is myometrium. Inside wall is endometrium. The uterus has 2 layers. And this endometrium wall is the place where the embryo implanted. Now, next question, discuss the social implications of IVF, means in vitro fertilization. Anika, I want to tell you one thing, and few of the commerce girl also, frequently you are talking in the grade 10A group. Yesterday I was busy with the work. When I came, I saw that there's a bulk of messages which is not related with the with the educational purposes or related with your class and all. Unnecessary, especially you are the common every time. You are sending emoji that you are talking your thought, you are sharing your thought, whatever, your pain thought, whatever you can, but do it in personal group, not in the, this is the class-wise group where all the teachers, principal, vice principals, we are included in this group. So please, this type of things, you know, that nobody likes. See, many teachers are giving complaint like that, and they are asking me, admin, why you are not controlling your girls. And I can understand you have grown up enough that this thing also I should tell, that don't use your personal, don't share your personal thought or personal messages in this educational group. So please maintain the discipline.
I'll definitely take care about it next time. I'll not write anything.
Please,
sorry.
yeah. And no emoji, this thank you, yes sir, no sir, please. Reduce this all. And many students are not getting their important messages. Suppose today grade 8, they didn't get my notes. They're telling me, because bulk of messages they are sending, especially the girls group. Boys groups are not like that. Continuously, they are each and of every students are sending, they are replying their messages. Sometimes they're each other also conversation going on. So what happened in between your own documents, you are not getting.
Okay.
And this public group posting the emoji, it's like crime, right? Why? This is the educational purpose. You are not allowed to send this kind of things. And this conversation between you and your friends, you use your personal group. Now, girls, discuss the social implications of in vitro fertilization. You know this thing. In vitro fertilization, some people support, some people don't support. because this in vitro fertilization allow infertile couples or the single parents. See, nowadays situations are like that. Many parents, they are not married, but they want a child, single parent. So even the surrogate mom also, you heard about this all, but that according to the religious and all, that is not supported actually. So the sperm is from one person, egg cell is from one person, the child is growing in another person's body, taking the food from that person's blood. So the purity of the birth is now questionable. So this in vitro fertilization also not having this kind of problem, but there is a question about this also. So it is good for the that infertile couple, that's true, or the parent, those who have single, but they want to have a child. But in vitro fertilization, it's not treat infertility successfully every time. If any couple are thinking that with this IVF, they will get the child, that is wrong. It will success or it will not. Sometimes there are many other factors impacting on them. And sometimes it causes multiple birth. To increase the rate, doctors are implanted 2, 3, maximum 3 that embryo. Some of them, sometimes 3 of them has been gone. As the treatment is very expensive, the couple has to go for several times the procedure for this implantation process. So this is very expensive and time consuming and makes the couple tired also. Sometime after implantation of the 3 embryo, 3 of them came as a child, successfully delivered 3 kids. So multiple birth is another problem, right? So this fertility treatment, first of all, is very expensive, creates multiple birth. And there is a religious and ethical issue about in vitro fertilization. And because many people do not support the waste of embryo, they are forming many embryo outside, and from the embryo, the healthy and perfect embryo, they are keeping in the uterus for the implantation. But what about the unused embryo, girls? Every embryo is a potential human being, right? So this is the ethical issue, the religious issue also, that sometimes, you know, that these embryos are using for stem cell therapy and all, stem cell research, but you are killing the human being. That is the main point. So the social implications of IVF is clearly you have the notes in your worksheet. But for the 4 marks, you can write like that: IVF allow the infertile couples or single parent to have the children, but IVF may not treat the infertility successfully, and it lead the multiple birth. In this infertility treatment is very expensive. There is a religious and ethical issue about the IVF treatment because many people do not support the waste of embryo, and this embryo also, this unused embryo also used for stem cell research or stem cell therapy. So this is the first question. We will be going to the second question. See, girls, second question, if you look at this picture, there is a big question, 6 mark. Pectinase is an enzyme used in the production of fruit juice. Describe in detail how enzymes function using pectinase as an example. Girls, this question, you need to write every points about the enzyme to get the 6 mark. All the enzyme properties you are going to write. After that, you will use pectinase as example, then you will explain the enzyme function. You just listen what I'm telling. Enzymes are proteins, and all the enzymes are specific. They can be used again and again and become unchanged after the reaction. These are all the properties of enzyme, right? So as they're asking you, detail how enzyme functions. So you are using also. So they can be used again and again and become unchanged after the reaction. Enzymes do not start or stop the reaction. They just speed up the reaction, which lowers the activation energy needed for the reaction and create successful collision. All the enzyme has active site. Someone is making sound. All enzyme has active site. If the shape of the substrate and the active site fits together, they form enzyme substrate complex. Enzymes has optimum temperature and pH. Too much heat and incorrect pH denature the enzyme. Substrate pectin, now we will come back to the pectin. Substrate pectin found in the cell wall of the fruits, which hold the cell together. So pectinase break down the pectin and the fruit juice can extract easily. So volume of the juice will be more and it will be more clear and cheaper. So girls, this is the way when they will ask, see, the notes you have in the pectinase enzyme is very small, right? Maybe it could be for 2-3 marks, but they're asking you detail how enzyme function. So all the properties and how the enzymes are functioning, these all you will explain. End of the answer, you will use your pectinase function as an example.
Miss?
Yes, girls, yes.
The enzyme part I can write by myself, but could you please dictate from the pectinase part?
Pectinase also you have notes.
No, the cell wall stuff, it's not there in our
It is
notes.
also in the notes. Girls, girls, you have this, right? Biotechnology notes you have, right?
No, biotechnology is there, but it's not written specifically that pectinase
Yes,
comes
it
from
is
cell
written.
wall.
Yes, it's written. You could ask other girls, or you can make the answer.
So, Miss, can you just repeat from there? Just
yes, you just listen, and that's what I'm telling. That's here. The pectin is a substrate, and enzyme is the pectinase.
yes.
Pectin is a substrate which found in the cell wall of the fruits, which hold the cells together. So pectinase enzyme break down the pectin. So the fruit juice can extract easily. The volume of juice will be more clear and cheaper. This is and it is in the biotechnology notes.
Okay.
So first you will explain the detail of the enzyme, then you will use the pectinase enzyme function. Now, next question. An experiment to test the effect of the size of apple pieces on the activity of pectinase was performed by a group of students. Some of their apparatus are shown in figure 2.1. They have used here one measuring cylinder, one funnel, and one filter paper. Now, girls, see.
Stopwatch.
Yes, dear.
One stopwatch.
Yes, one stopwatch also there. Describe how the student could use the measuring cylinder to obtain the accurate measurement of the volume. This you know, right? First, you will place the measuring cylinder on a flat surface. Remove the funnel and make sure all the drops have fallen in the bottom. Read at the eye level to avoid the parallel, that error of parallax. Okay. Now, that bottom of the meniscus also you can write. After filtering, you have to remove the funnel. So there are 2 marks. So first point, you will write, place the measuring cylinder on a flat surface, then remove the funnel, and make sure when you are going to remove the funnel, the bottom part of the funnel, the boundary, look at this arrow, the bottom part, sometime when you're removing, the drop will fall down, right? So wait, so all the drop has to fall in the bottom. And then read the eye level to avoid the parallax error. Got it, girls? I have given you the 3 points. First, you'll keep it in the flat surface, then remove the funnel, but make sure all the drops in the bottom part of the funnel will fall down in the bottom of the that cylinder. Then measure the reading in the eye level, or you can read the bottom of the meniscus also.
Miss, could you please repeat the answer again? I'm sorry.
Anika need all the answer, right?
No, repeat. Yeah. I don't need it. Like I didn't understand. No.
You don't understand. Cylinder you will keep in the flat surface. And then this funnel you will remove. And when you remove, make sure that the bottom of the funnel, because it is inside that liquid, right? It will fall drop by drop. So wait, so the drop will fall on the cylinder, then remove the funnel. Then measure the liquid read the eye level to avoid the error of parallax.
Thank you.
Welcome. Now, next question. The student added 1.5 cubic centimeter of pectinase solution to pieces of apple in a beaker. Someone is, could you please mute your microphone?
Sorry, Miss.
Who is making this sound? Like the sound I'm getting is like someone is cleaning or washing like that. Anyway, the student added 1.5 cubic centimeter of the pectinase solution to piece of apple in a beaker. Remember, you have taken one beaker you kept 1.5 cubic centimeter pectinase solution. The pieces of apples are also there. Then you pour the mixture into a funnel. That they found that it took 10 minutes to collect 19 cubic centimeter juice. You understood, girls, that they have kept the apple juice. Okay, someone is giving me the messages. It's okay, Zaina. Now, they found it took 19 minutes to collect 19 cubic centimeter juice. So they have kept the apple in the beaker. Now they have given 1.5 cubic centimeter Pectin Is, and then they poured the mixture into the funnel. Finally, they found that it took 10 minutes to collect 19 cubic centimeter juice. Calculate the rate of enzyme reaction. Girls, 19 divided by 10, or 19 divided by 1.5, then divide with the 10, but better to 19 divided by 10. 19 divided by 10, it will be 1.9, it will be 2 cubic centimeter per minute. So you know this thing. And then after that, next question, see that here they're asking, they have given you that a few data. The student performed 4 experiments using different ways to prepare the apple juice, right? The same total mass and the type of apple was used in each time. Okay, different experiment. First, they have used that 0.5 cubic centimeter apple cubes. Second, they have used 1 cubic centimeter apple cubes. C, they have used whole peeled small apples, whole unpeeled small apples without chopping or without cutting into small pieces. So predict and explain with which experiment in A, B, C, D would result in the fastest rate of reaction. Girls, it will be A, right?
Because it's the smallest piece of cake.
Yes, because it's the smallest pieces means more surface area. So A, because 0.5 cubic centimeter apple cubes, because A is the smallest cube and have the large surface area so enzyme work faster to give the result quickly. This also you know. Actually, girls, questions are not hard. The problem is, what is our student problem? Sometimes they couldn't, understand that how much they should write in every question. They see, this everything you know. Actually, if you memorize the notes, answer will be common. For all the answer, you will get it common. But the problem is sometimes you didn't understand how to combine the question with the notes. So that's why you need to practice past paper more. If you look at this diagram, see, the length of the small intestine was measured in 4 types of mammals. The result they have given. Length of the small intestine in insect-eating bat is 19 centimeter, and the length of the small intestine relative to the body mass, 2.30 centimeter per gram. Domestic cat, domestic cat is 104. And the body relative to the body mass is 0.05, the rat and the human. Look at this human. So length is so much, but compared to our body mass is 0.01. Now, what is the question? Use this information in table 3.1 to compare the length of the small intestine for the 4 mammals. So these are the 4 mammals. See, by using the information, you just need to compare. No need to explain. So easy question. Just compare. How to compare? See, human has the largest 552 centimeter small intestine, and insect-eating bat has the shortest 19 centimeter small intestine. Length of the small intestine and the rat and the domestic cat are very similar, 98 to 104 centimeter. But there is a negative correlation between the length of the small intestine and the length of relative to body mass. According to the relative body mass, bat is much larger than the mammals. So you can write that there is a negative correlation compared to the length with the relative to the body mass, because insect, it has the lowest length, but it has the highest relative to the body mass. Human has the highest length, but it has the least that relative to the body mass. So this is the way you just compare the result. It's not a problem, girls. So these questions are nowadays also, it is coming in 19 also, this type of this pattern of questions came. So this question, this is a diagram showing the short length of small intestine to a mammal? See the outer surface, inner surface, and the lumen. The function of a small intestine is absorption. We know these all, and they have a special structure of this villus and microvilus, dense network of blood capillary, more mitochondria. You know these all. And they are producing that hydrochloric acid also. You know these all. Describe how a molecule of glucose passes from the lumen of the small intestine into the blood. See the question. From the lumen of the small intestine into the blood. See, now go back to your digestive system function. From the lumen of the small intestine, glucose molecule moves through the epithelial cell of the villi. And then it moves into the capillary across the cell membrane by diffusion or by active transport. They use energy which has come from the cellular respiration for the movement against the concentration gradient and then pass it into the blood. Actually, same thing as it is 3 marks. You need to write that from lumen, the small intestine, the glucose molecule moves through the epithelial cell of the villi and microvilli. Then it moves through the capillary across the cell membrane. It might be by diffusion or active transport. If they use active transport process to move against the concentration gradient, they use energy which has come from the cellular respiration. So girls, this is the question. Okay, the answer is actually 1 line. As it is 3 marks, you have to make the additional, the process also you have to explain to get the 3 marks. This is another pattern of writing answer. Because whenever the main answer is 1, the relative part you need to explain to get the full marks. So next question. So see, this measurement were taken of an inner and outer surface area of 2 parts of the small intestine for the 4 mammals: insect-eating bat, domestic cat, rat, and human. So the ratio of the inner surface area to the outer surface area. Look, insect-eating bat has the highest ratio. Look at the human and all. Now suggest which mammal has the most villi per centimeter of the small intestine?
Insect-eating bat?
Yes, because see, inside the ratio inner surface, surface area is more than outer surface. Even the duodenum is 283 and ileum is 54. So it has more the villi inside. So it has most the villi per centimeter. That answer will be insect-eating bat. Thank you. Now, next question. The duodenum is more effective than the ileum at absorption. Use the information. See, duodenum, look at the ratio. In the duodenum is higher than the ileum, right? So now they're asking you use the information to explain why duodenum is more effective than the ileum. See, by looking at the diagram, you will explain in this way. Ratios are higher in duodenum because duodenum contains more villi and more microvilli, which increases the surface area in the duodenum than the ileum. For human, the ratio of the inner surface area than the outer surface area is 7.1 in the duodenum and 3.1 in ileum. So absorption will be more in the duodenum than the ileum. You will just explain in this way. Whatever you are saying in the table, you will explain in this way. I'm telling this answer again. Ratios are higher in duodenum because duodenum contains more villi and more microvilli, which increases the inner surface area in duodenum than ileum. For human, the ratio was 7.1 in duodenum and ileum was 3.1. So the absorption will be more in duodenum than ileum. So this is the answer. Just write from the table. Now, next question. See, the bile is released into the small intestine from the gallbladder. You know the bile produces in the liver. It goes to the gallbladder, stored over there. Through the bile duct, it comes to the duodenum. Outline the role of bile. Girls, bile is an enzyme. Is bile is working as an enzyme?
Yes. No, it doesn't. It's just a juice, digestive juice. Wait, it does work as an enzyme.
Thank you. Bile do not contain any enzyme. It is a yellowish green substances. Bile is a yellowish green alkaline water liquid, you can say. It doesn't contain any enzyme. It just breaks down the large fat globules into small droplets,
Emulsification.
which we call emulsification. Yes, bile increases the surface area, making it easier for the lipase to digest them into that fatty acid and glycerol. It also helps to neutralize the acidic mixture from the stomach, because due to the presence of hydrochloric acid, the food molecules are more acidic, the kind, right? And this bile
makes
work on it,
alkaline.
yes, to provide this, they also neutralize the acidity, because the trypsin, which has come from the pancreas, couldn't work properly in this acidic condition. So girls, this answer will be bile is a yellowish green alkaline watery liquid. It doesn't contain any enzyme. It just breaks down the large fat droplet into small droplets, which call emulsification. It increases the surface area of the fat molecules and make it easier for the lipase to digest them into fatty acid and glycerol. It also helps to neutralizes the acidic mixture from the stomach and provide the alkaline medium for the pancreatic enzymes, so that the other enzyme can work well. So this is because it's 4 marks, you have to explain. Always check the amount, check the number in every question, so your explanation will be according to that question.
Miss,
now,
provide
girls,
alkaline medium for proteolytic enzyme.
It provides alkaline medium for that pancreatic enzyme, you can write.
Oh, pancreatic enzyme.
Even the pancreas also secretes sodium hydrogen carbonate, which also neutralizes the acidity. We have 1 minute 30 seconds. I'll be coming back with the new link, girls. But before that, I want to ask you. First, if the questions are like this, if tobacco smokes is made up of over 7,000 chemicals. See, one cigarette contains 7,000 chemicals. I told the smoker person that is it possible for them to show me one example which is good about the smoking. They are telling that if I'm not smoking, I'm not feeling hungry. Okay, I got headache. But do you think this is all actually addiction, right? There is no relationship between that smoking with the hungerness, smoking with the that headache. They are addicted. That's why if they're not taking the smoking, if they're not smoking, they're getting this cigarette. They are not feeling to eat food. So many problems they are having. These are for the addiction. So girls, I'm coming back, okay, because our meeting time has over. Less than a few seconds only. I'm coming back, okay?
Okay.
Yes, welcome back, girls. We are going to start our second session. Yes, Anika, what was your question? Just share with us.
I've heard this fact that cigarette prevents Alzheimer, like smoking. Is it true?
No, I didn't hear this kind of evidence still now. How? Cigarette contain 7,000 chemicals. None of them are good for the health. See, now, so this nicotine is one of the component of tobacco smoke. Alzheimer's, that cigarette, there is no relationship. Now, explain why nicotine is a drug. Girls, instead of nicotine, any types of substances they are keeping and they're asking you why this is a drug, what will be your answer? In the place of nicotine, if they're asking anything, okay, any types of, substances extracted from the plants or animals, they're using that term, they're asking you that explain why this is a drug. So in that cases, you are going to use the definition of the drugs. You know the drugs. What is the definition? You have learned from your notes. You learned that the chemical drugs, these are the chemical substances taken into the body, which modifies and affect the chemical reaction, and some of them are causes addiction, and also it gives some symptoms. So this is the definition you have learned from the notes. This definition you will mix with the question. How? Explain why nicotine is a drug. You will explain in this way. See, nicotine is a chemical substance. Exactly drug definition you are mixing with this. Nicotine is a chemical substance taken into the body through the tobacco smoke, and it modifies or affects chemical reaction into the body. It is highly addictive and causes severe withdrawal symptom when stopped. So see that in this way, you can also explain from the definition, from the different question answer, from the explanation, you can make the answer of the past paper question. Maybe you can, at a glance, you may think, why nicotine is a drug? This question you don't have. And that's true, but you have what is drug, right? So that same answer, you will just mix with the nicotine to get the answer. Next question. Describe the effect on the gas exchange system of the following component of tobacco smoke. First, they are asking about the carbon monoxide. Girls, this is the poisonous gas, right? You know why?
Combines with hemoglobin and forms carboxyhemoglobin, which is toxic, and it doesn't allow oxygen carrying capacity in blood. It stops.
Yes, so your body cell deprived with the oxygen, which makes you anemic, right? And also the fatigue will be more. You will not get the energy. So 100% right answer you have given. See, carbon monoxide is a poisonous gas. It diffuses from the lung into the blood and combined with the hemoglobin form carboxyhemoglobin. It means there is a less oxygen carried and the body cell will deprive with the oxygen. And now tar. Girls, one special term I gave you in the notes about the tar, right? Tar contain many different chemicals and some of them are carcinogenic, which causes cancer, right? Like lung cancer. Yes, because you know that tar stick and damage the cilia. And alveolus produce more mucus, which causes infection and reduce the gas exchange process. Now, COVID-19, what it is doing, you know, it is just destroy your lung. So your lung couldn't supply oxygen to your organ, and all the organ need constant supply of the energy, and that energy has come from the aerobic respiration. By using oxygen, they are breaking down the food molecules to provide that oxygen. So if the oxygen supply has been stopped, so they will not be able to provide energy to your organ, because your organs are working every time. Metabolic processes are going on every time. If your brain cell do not get oxygen 3 minutes, it will stop working. You will go either coma or you will die. So this is the reason that lung functions are very important, girls. All the organs functions are important, but lungs, see that if your body cells are not getting enough energy, they will not work. This is a different types of growth. Girls, there are four lines. Now, a student, the study compared that the percentage of men and women aged 35 to 54 years who smoked cigarette. The annual death rate caused by the lung cancer also recorded. So first graph, they have showed the percentage of smoking, the way it is increasing men and women. And the first question, see, girls, use the data shown in figure 4.1 to compare the percentage of men and women who smoke cigarette between 1950 to 1998. Here, you just compare the data. No need to write the reason. So how? Look at the graph, girls, everyone. Look, the first graph, okay. More men smoked between 1950 to 98 than women, right?
Yes.
Both percentage decreases between 1970 and 1998. Check. Overall drops in man is more from 82 to 38. Check the first data and the last data in 1950 and 2000. So overall drops in man is more from 82 to 38 than women from 40 to 28. So I have given already 3 data here. And now you can give the direct data like that. In 1950 to 1970, the percentage of smoking in man was decreasing, but the percentage of smoking women was increasing. Right, girls? Before 1970, there is a large differences then between men and women. Then after 1970, the differences become less. The maximum percentage of smoking for women was 50% and the man was 82%. So this is the way. If you want to get 4 marks, you have to explain in this way. I want to tell you again this answer. More men smoked between 1950 to 1998 than women. Both percentage decreases between 1970 and 1998. Overall drops in men is more from 82 to 38 than women from 40 to 28. In 1950 to 1970, percentage of the smoking in men decreasing and the women increasing. Before 1970, there is a large differences larger differences. Then after 1970, maximum percentage of smoking for women was 50% and for men was 82%. This question was easy, just compare from the data. Next question, see. Use the information, both the graph, because this is a bit confusing, from both the graph in figure 4.1 to discuss the link between smoking and lung cancer. Go back to the diagram. Now here you need to understand the link between smoking and the lung cancer. So we will compare both the diagram for both men and women. See, number of death by lung cancer shows similar trend as percentage of a smoker, right? If the percentage of a smoker is more, death is more. If the percentage is less, death is less. So you will give the answer like that. Number of death by lung cancer shows similar trend as percentage of a smoker. There was correlation in both men and women. In 1950, the percentage of smoking in men was 82%. And the annual death rate is 52%. Got it, girls? From the graph, you can understand. And percentage of that in women, 1950, the percentage of smoking was 40 and the death was 8. So the more percentage, more death, even between the men and women. So you will describe both the graph in this way. Death rate of the man is decreasing, and in 1998 it becomes 19. But death rate of women is increasing, and at 1998 it was 12. So here a bit contradictory relationship they are showing. You just stated, explain with the graph what they are showing. So girls, this is the answer of this question. If I'm going to the next question, this answer you know, explain why it is recommended that the pregnant women do not smoke. See, nicotine and carbon monoxide can cross the placenta, right? And can enter into the baby's blood.
For carrying blood.
Yes, this increases the risk of miscarriage, premature birth, or low birth weight, and also deformities. So it reduces the oxygen availability, that availability to the fetus, and causes the brain damage. It may be the reason for the brain damage of the fetus. It increases the risk of infection, and also increases the lung couldn't work properly due to this smoke. So girls, you just write the 3 points like nicotine and carbon monoxide can cross the placenta, enters the baby's blood, which may causes deformities in the child and causes the fetal brain damage. It also increases the risk of miscarriage, premature birth, low birth weight, etc. So you will get 3 marks. Now, next page, 14 is the blank, 15. Girls, describe the structure of DNA molecule. No need to explain, this is direct notes. DNA looks like double helix, spiral ladder-like, 2 sides of the frames, alternating sugar phosphate connection with the 4 bases, adenine with thymine by 2 hydrogen bond, guanine joined with cytosine by 3 hydrogen bond, and in between there is a pair of nitrogen base present. You know this thing, direct notes. Functions of the gene, that codes for a protein, right? Functions of mitochondria, aerobic respiration happens in mitochondria to release energy. Now, some scientists think that the mitochondria evolved from bacteria because they are the similar size and structure. Bacteria belong in the prokaryote kingdom. Give 2 features of all prokaryotes.
Contains genetic material and has protein in it. No, not really. Has nuclear.
They have 2 nucleus. They have mitochondria, do not have 2 nucleus. They have loops of DNA, right? Plasmid, you can say. They're unicellular. They do not have nuclear membrane. They do not have actually proper nucleus. They have genetic materials, you can say.
I remember
mitochondria, they do not have mitochondria. They are unicellular. They have loops of DNA, which we call plasmid.
Yes, miss.
And this one, if you notice, see, by using this information, they are asking to find out use the stable to show that, look, first, the most close E and G. After that, which one is very close to G and E? So you have to think that the bottom one has been divided. After that, no branching point in the bottom 2. So if you go here, look at this A and F. Look at this A and F. So after branching point, it did not come. So in bottom here, it will be A and F. E and G, if you go back again, look, E, okay, E and G. If E are, that's G, and E is here, that they are showing you the DNA base sequences, the number of the differences. So the E and the G, after G, then they are showing you E, and then it will be C. Look, 6, and then that if it is 28, then with, look, this is 6, and species E and G is 6. After that, this species will be with the species C.
Miss,
Look,
how we are
species
doing
C.
this, I'm not understanding.
Yeah, that I can understand. This question is actually, look, I need both the pages in front of you to make it clear. DNA, this table shows the number of differences between DNA sequences. So if you notice, there is no differences between mitochondria A and B, but species B, okay, there is no differences with the species B, but with C, species B has the differences, the DNA base sequence differences 18. Then with D, 12, with E, 17, with F, 26, with G, 24. So they are asking with C, species C, that the differences of G is 14, and with C, the differences of E is 10. Okay, so if you look at this question, both the pages, it should be together, then it will be easy. So if you are looking G and E, after that, the question is, look, place the letter for each species or the mitochondria in the box next to the correct branch of the classification. So the less differences will be close, right, girls? So if you're saying that species G and species E, so species G and species E, 6. After that, which one is related with species G and E? Less. So you can see that this is coming, look, species G and look, species C, there is no differences with C, no difference, that 19 difference in E, but E is 10. So after E, it will be C. Understood?
Miss, what is this happening? I'm not understanding.
How to make clear? I told you, keep these two pages together. You have to find out the less differences. I told you species G and E has 6 differences.
So the less difference,
yes,
we find out by looking at the number.
Yes, looking at the number. The less number means the less differences. The more number means more differences. So they have given you the question. They are showing you E and G. Next to which branch will come with E and G. E and G, you just look, that's E and G, the C is coming. Because look at species C. Look, with species C, E is 10 and G is 14. So if it is written G and E, after that C will come. And after C, D will come, because after 10, 19 with that sequence. And then it will come to the last is B. Because they're asking you species G and species E in relation of these two. So no problem, you just try, circle all the differences and inform me if you don't understand next class. Such is why using DNA sequences is useful, because to identify this, the species, those are very similar and more accurate. It's easier, and that you can do it quickly. Question number 18, girls, is very easy. Name one feature of the dicotyledons dicot leaves that distinguishes from monocot. You can write
dicotyledons have 2
why the leaves and organ, no need to tell. Balance equation of the chemical equation of the photosynthesis, no need. Now they have shown here one image. Look at this diagram, and they're asking layer J. What is layer J? Girls, look at the layer. J is the palisade mesophyll layer, right? Xylem. Palisade mesophyll layer.
Palisade mesophyll, yes. The leaf structure.
This is the leaf structure.
Yes.
So the J is palisade mesophyll. And what is the adaptation? Tightly packed, oblong shape, and contain lots of chloroplast.
Yes.
Now, layer K. What is layer K, girls? Look at the K.
Upper
That is
epidermis.
upper epidermis.
Upper epidermis.
What is the adaptation?
They don't.
Transparent?
They have few chloroplasts. They have no chloroplasts.
Transparent, right? And allow the light to
Cuticles.
pass through it. Yeah, no chloroplast,
Oh, okay.
transparent,
Gas exchange.
and allow the light to pass through. Now, layer L. What is L?
Spongy mesophyll.
Spongy mesophyll.
Good. These are loosely packed, have air spaces to allow the diffusion of oxygen, carbon dioxide to and form the cells, like gas exchange. Now,
Yes.
the last question of this chapter is, plant need nitrate ions to grow. See, girls, these also clear notes, right? Plant absorb nitrate ions from soil. Nitrates are usable source of nitrogen and needed to make amino acids, and which they are that are using to make the protein, because proteins are needed to make DNA, RNA, and to make new cells, which causes their growth. Without nitrate ion, plant growth will be less. The leaves will be yellow in color, the stunted growth, and they will not produce sufficient, the yield or amount of crops will be less due to the lack of nitrate ions. Someone is drawing. Okay, girls, this is our today's that past paper. Now, want the question. So tomorrow, as you want to do this February, March, I will give you that February, March question today PDF form. So few pages we have completed, right? Few pages I did not do. So after doing these few pages, we will have time, right? Shall we discuss one paper 6?
Okay, miss.
So that paper 6, which variant that you want to do paper 6 from 19, or you want to do paper 6 from 17?
Miss from 17?
Okay, 17, then that I actually, that many past paper actually I have done with you, right? So 17, which one I didn't do? You need to inform me that. Any idea that 17, that which one we didn't do?
Did we do 6-1 or 6-2 like of October, November?
And then 6-3 May, June, did we do 17, 6-3 May, June?
6-3, I don't have it in my past paper. I have to download it.
I'll give you the that question, no problem. So no answer from other girls. So okay, better I'll send you the PDF, right?
I don't remember whether we did it or not.
Okay, no problem. Better choose one that topic. February, March 17 past paper variant 4.2. I'll send you the PDF, and also that we are going to solve one of the paper 6 past paper, that paper 2, that 6.2 variant you want, or that 6.3 variant you want.
Paper, I don't know, miss. Let others decide. Like, it's just me talking. That's a little weird. You know.
I don't know. Any paper, miss, you can say the year.
Okay. Then I will just send you the PDF, that 6.2 or 6.3 variant, anyone. We will solve that one, okay? So you want paper 6 from 17 variant, then I will not upload 2 PDF, okay? The February, March, you people just get ready by yourself. I will upload only the paper 6, okay? Okay, thank you, girls.
What is it again? I'm sorry. What should we get ready with us?
February, March 17 variant will be, you will download by yourself. But in the group, WhatsApp group, I will give you the PDF of paper 6.
February, March, the things I was talking about, the last variant, right?
Mm-hmm.
Okay.
That's the first week we did.
Yes,
that
it was
the
just
combined class. No problem. So how many question answer we didn't do? I will discuss February, March, and then I will start paper 6. That's 17. Any variant, I will upload it, the PDF. Okay. Okay. Thank you, girls. And this is our end of our class. Your next class is going to start right now. Thank you, girls.
Thank you.
Thank you.
Thank you.